Hi everyone, and welcome to this session where we're going to look at ketone bodies, that is the synthesis and also the breakdown of ketone bodies. And so by the end of this session, we'll be able to describe the metabolic conditions and also hormonal signals that promote ketone body formation, outline the biochemical pathways of ketogenesis and name the major ketone bodies, as well as be able to describe how ketone bodies are transported, utilized, and converted back to acetyl-CoA, and relates the physiological and clinical significance of ketone metabolism. So ketogenesis is a synthesis of ketone bodies, and it occurs in the liver. And we see that in a well-fed state, glucose is abundant, insulin levels are high, and liver prioritizes glycolysis and glycogen storage. However, when the body enters a state of carbohydrate scarcity, for example, during fasting, prolonged exercise, or even starvation, the metabolic landscape changes. And we see that the blood glucose levels fall, insulin secretion decreases, and glucagon and catecholamine levels will rise. And so this hormonal shift signals adipose tissues to begin lipolysis, releasing large amount of free fatty acid into circulation. The liver will then take up the fatty acid and channel them to beta oxidation, producing high quantities of acetyl-CoA. Now, it is important to know that under normal conditions, acetyl-CoA enters the citric acid cycle, but during low glucose level, the cycle slows down because oxaloacetate is diverted towards gluconeogenesis. And so with the TCA cycle capacity reduced and acetyl-CoA accumulating, the liver must find an alternative fate for the surplus. And so this is the moment when ketogenesis becomes essential. So inside the mitochondria, excess acetyl-CoA is condensed to form acetoacetate, β-hydroxybutyrate, and also acetone, which are collectively known as ketone bodies. Now, the ketone bodies are water-soluble, and therefore they're easily transported in the blood and serve as crucial alternative for fuel for extrahepatic tissues, including the skeletal muscles, heart, and also during prolonged fasting for the brain. So as we've seen that the hormonal environment is essential to ketone production. And so let us look at when does ketogenesis occur in detail. During fasting and starvation, we will find that blood glucose will fall, and therefore the insulin levels will also decrease, whereas the glucagon levels will increase. And this will lead to activation of hormone-sensitive lipase in the adipose tissue, which will release free fatty acid from TAG into blood. So the liver will take up the free fatty acid and perform beta oxidation, producing large amount of acetyl-CoA. On the other hand, because oxaloacetate is diverted to gluconeogenesis during fasting, the TCA cycle will slow down, and therefore acetyl-CoA will accumulate. The liver will then convert the excess acetyl-CoA into ketone bodies, and this occurs especially in the brain after 2 to 3 days of fasting. Prolonged exercise will also result to formation of ketone bodies, and you see that long duration exercise will cause depletion of muscle glycogen, and this will lower blood glucose. This will lead to low insulin and high catecholamines, and thereby increase lipolysis, which will increase fatty acid mobilization, and high rates of fatty acid oxidation in the liver will result to formation of acetyl-CoA. And therefore, the liver will shift to ketone body production to supply fuel to muscles and brain during prolonged exercise. In regards to low carbohydrate, we see that an intake of low carbohydrate will mimic fasting because insulin will stay low, glucagon will remain high. And this will lead to lipolysis and therefore continuous formation of acetyl-CoA. And also they will be accompanied by reduction of oxaloacetate availability for the TCA cycle. So the body will enter nutritional ketosis, providing ketones to maintain blood glucose and support brain energy, and therefore spare glucose that is needed for energy in the brain. Poorly controlled type 1 diabetes mellitus will also lead to ketogenesis, and this is one of the most severe and dangerous triggers of ketogenesis. This is because absence of insulin will lead to massive and inhibited lipolysis, and therefore extremely high levels of fatty acid will be delivered to the liver, resulting to overproduction of acetyl-CoA from beta oxidation. So with no insulin, glucose cannot enter cells, and therefore the cell will think they are starving despite high blood glucose. And also we see that oxaloacetate will be used for gluconeogenesis, and this will also affect TCA cycle. So there will be accumulation of huge levels of acetyl-CoA, which will lead to excessive ketogenesis, producing diabetic ketoacidosis. And this is a pathological state. Alcohol ketoacidosis, and this is as a result of heavy alcohol consumption, especially with poor food intake. And it will increase NADH from alcohol metabolism, and therefore NADH NAD+ ratio will inhibit gluconeogenesis and also the TCA cycle. As a result, it will lead to low glucose because low insulin, high glucagon, which will increase lipolysis, and therefore will have massive fatty acid delivered to the liver. In addition, we'll have blockage of the TCA cycle because of absence of NAD, which will be taken up by alcohol metabolism, and therefore there will be a buildup of acetyl-CoA. So this will lead to formation of ketone bodies, resulting to alcohol ketoacidosis. So let us look at the reaction sequence that leads to formation of ketone bodies. And the first reaction, or the first step in ketone body formation, is a first committed step where we have condensation of two acetyl-CoA molecules to form acetoacetate-CoA, a reaction catalyzed by thiolase. Now, the second step, through the catalyzation effect of HMG-CoA synthase, it's a formation of hydroxybutyrate-methyl-glutaryl-CoA, that is HMG-CoA. And this is a rate-limiting enzyme of ketogenesis, that is HMG-CoA synthase is a rate-limiting enzyme because it is highly expressed during low insulin state, and therefore it's a key regulatory point in this pathway. And it condenses acetoacetate-CoA with another acetyl-CoA to give us HMG-CoA. HMG-CoA is then cleaved by HMG-CoA lyase to yield acetoacetate and also acetyl-CoA. So acetoacetate is the first true ketone body to be formed, and it has two fates. The first fate, which is characterized by beta-hydroxybutyrate dehydrogenase, will lead to the formation of beta-hydroxybutyrate. And the direction of this reaction depends on the mitochondria NADH NAD+ ratio. So when NADH levels are high, in case of fasting, heavy alcohol intake, intense beta oxidation, the reaction shifts towards formation of beta-hydroxybutyrate. And this explains why ketone bodies often predominate in ketosis and also ketoacidosis. The other alternative fate is a spontaneous decarboxylation to form acetone. This is a volatile ketone responsible for the characteristic fruity breath odor seen in ketosis and also ketoacidosis. Both acetoacetate and beta-hydroxybutyrate will be transported into the bloodstream. Remember, they are water soluble, and they will travel to peripheral tissues such as the muscles, kidney, heart, and ultimately to the brain during prolonged fasting. So let us look at ketolysis. And this is a degradation of ketone bodies. And have you seen that the ketone bodies are produced in the liver and then they are transported to the peripheral tissues through circulation, where they are utilized for energy production. This is very important because we see that the heart and the kidney, in most cases, they prefer to use ketone bodies rather than glucose for energy. And during prolonged starvation, the brain derives most of its energy from ketone bodies to conserve glucose. It is important to note that the liver is unable to use ketone bodies as a source of energy or as a source of fuel to produce energy due to the lack of succinyl-CoA acetoacetate-CoA transferase, that is a very important enzyme in the degradation of ketone bodies. And so let us look at what is the fate of beta-hydroxybutyrate and also acetoacetate. In the first step, beta-hydroxybutyrate is converted to acetoacetate by a dehydrogenase, that is hydroxybutyrate dehydrogenase, and you see that NAD+ is a hydrogen acceptor. However, we have a minor route to form acetoacetyl-CoA, and beta-hydroxybutyrate can be directly activated by a synthetase to give or to form beta-hydroxybutyryl-CoA, which is followed by a dehydrogenation reaction, where we see that beta-hydroxybutyryl-CoA will be converted to acetoacetyl-CoA, a reaction catalyzed by dehydrogenase. Acetoacetate formed in the major route is then activated, a reaction involving a transfer of CoA from succinyl-CoA, a reaction catalyzed by succinyl-CoA acetoacetate-CoA transferase, also known as the ketoacyl-CoA transferase. And this is the enzyme we said that the liver does not have. And the product of this reaction is acetoacetyl-CoA. So remember we talked about the minor route. So the beta-hydroxybutyrate can be converted directly to acetoacetyl-CoA through the actions of a synthetase and also a dehydrogenase. Acetoacetate can also be activated to acetoacetyl-CoA through a reaction catalyzed by acetoacetyl-CoA synthetase, and this occurs in presence of ATP, magnesium, and also CoA. So acetoacetyl-CoA formed can then undergo cleavage, and this is catalyzed by a thiolase, and it gave rise to two molecules of acetyl-CoA. And so acetyl-CoAs are then subsequently oxidized by the citric acid cycle to release energy in form of ATP and also NADH, NADH, and also the FADH2, which now progresses on to the oxidative phosphorylation. When it comes to acetone, we see that utilization of acetone by extrahepatic tissue is low, and therefore it is removed by excretion in urine or as carbon dioxide through the lungs. Now, it's important also to mention that ketogenesis is also under regulations, and there are various ways. The first one is mobilization of free fatty acid from adipose tissue will control the rate of ketogenesis. Here we see that any condition that increases mobilization of fat will increase ketone body formation. Liver carnitine acid transferase 1, that is CAT1 activity, will also determine the rate of ketone body formation. Under fed conditions, we see that CAT activity is inhibited by malonyl-CoA, and therefore it will decrease ketogenesis due to less acetyl-CoA. On the other hand, during starvation, CAT1 activities is high due to low malonyl-CoA, which will lead to conversion of TAG to fatty acid, and therefore they're transported to the mitochondria, where they undergo beta oxidation to release plenty of acetyl-CoA. ATP level also controls ketogenesis. So we see that more ATP levels favors ketogenesis, whereas low ATP level prevent ketogenesis in the cell. Hormonal imbalance, we see high levels of insulin will decrease ketogenesis, whereas high levels of glucagon will increase ketogenesis. We also saw that TCA cell activity is also determine the rate or regulate ketogenesis, because we are talking about availability of oxaloacetate. So if oxaloacetate is missing, that means we'll have more ketone bodies being formed from acetyl-CoA. So let us look at the clinical relevance of ketone bodies metabolism. The normal blood ketone body levels is about 1 milligram per 100 ml. However, under certain conditions, the rate of ketone bodies formation may exceed the rate of their utilization by peripheral tissues. And this will lead to ketosis, which is a physiological state in which production of ketone bodies is elevated above baseline, but it remains within a normal or a range compatible with the normal acid balance. And you may either talk of ketosis being hyperketonemia, that is elevated ketone bodies in blood, and the blood pH is usually normal or slightly reduced. And it's a laboratory measurement, but not a disease state. And it can occur both in physiological and also pathological conditions. In physiological hyperketonemia, the liver produces ketones during fasting, exercise, or low-carb diet, and the peripheral tissues efficiently use them. However, in pathological hyperketonemia, such as diabetic ketoacidosis or alcohol ketoacidosis, we see that the ketone levels are excessively high and can overwhelm the body buffering capacity. When you talk about ketonuria, it's elevated ketone bodies level in urine, and it occurs now when blood ketones level exceeds the kidney's reabsorption ability. So it's important to note that it occurs in starvation when you have uncontrolled diabetes mellitus, high fat diet, or when you have severe muscular exercise. And the main clinical symptoms are headache, nausea, vomiting, and finally, coma may occur in elevated level. However, under ketosis, it's also important to note that under this condition, ketone bodies serve as valuable alternative energy source for the brain, heart, and also skeletal muscles. And this will spare glucose for tissues that obligate glucose users, like the RBCs. Ketoacidosis, this is a pathological state where we have severe accumulation of ketone bodies and will lead to metabolic acidosis, that is low blood pH. And it occurs when ketone bodies or production of ketone bodies exceed the body capacity to use or even excrete them. And their formation in large quantities during starvation or high alcohol intake causes depletion of blood bicarbonate, and therefore this results to lower blood pH. Finally, we have hyperketonemia, and ketone body formation may be impaired in some diseases like the carnitine deficiency and also the hepatic CAT1 deficiency, resulting to low ketone bodies and therefore the hypoketonemia state. And so with that, we've come to the end of this session where we have looked at production of ketone bodies, their degradation, and also their medical importance in regards to their metabolism. Thank you.